So when we're talking about nucleic acid in this class, probably most often we'll be talking about the polymeric form, outside of talking about like ATP as an energy source, of course. So as we kind of build towards this polymeric version of DNA or RNA, the physical properties probably change. The solubility changes as we add more charge from all those phosphates, the polarity from the sugar, for example, and its melting point might also change due to the number and types of interactions that are happening in the polymer. Its chemical properties change. Its charge, as I mentioned, is probably getting quite negative due to all those phosphates, and therefore its reactivity might change. Of course, from your biology class, you've learned that as DNA or RNA polymerizes, it begins to store information due to the sequence of nucleotides. In terms of the structure of DNA, there's some kind of basic things that I am assuming you are maybe slightly familiar with from other classes. There is Chargaff's rule that kind of tells us that A and T are always found in equal proportion to C and G. Why this is, we'll see on the next slide. The structure of DNA was largely based on Rosalind Franklin's X-ray crystallography that showed DNA was a double helix, and this really formed the crux of our understanding of DNA structure. Jerry Donohue elucidated the correct forms of bases, so the bases were hypothesized to either be in the keto or enol form, words that might be familiar to you from organic chemistry. He was able to determine that the keto was the more stable version of the bases. Now, why Chargaff's rule? Why are A and T always found in equal proportion to C and G? Well, you probably already knew this, folks, but base pairs hydrogen bond with each other. A and T pair with each other because they can form 2 hydrogen bonding interactions between themselves. C and G base pair because they form 3 hydrogen bonding interactions between themselves. And so all of this information built towards our current understanding of Watson-Crick-Franklin base pairing and the structure of DNA. We know that the 2 strands are anti-parallel. So one is 5' to 3' that way, and the other is 5' to 3' in the opposite direction. They form a right-handed helix. So what that means is, if you take a quick look at my picture for a second, and this is my right hand, and I think the video is kind of backwards for you, so go ahead and put your right hand up. What it's telling you is the helix curves towards the right like this, and its directionality is upwards. Finally, they were able to elucidate that the bases occupy the inside of the DNA helix, and the phosphate and sugar backbone is on the outside. Finally, we know that these planar bases on the inside hydrogen bond with each other. Let's dive a little bit deeper into the structure of DNA. So there are 3 possible forms of DNA. B-DNA is the most common form found inside of cells. It has that anti-parallel arrangement that we discussed, and it is right-handed. There are 2 other major variants that we're not going to focus on too much outside of a few points here. So A-DNA, it's a bit wider. It's found in the polymerase active site, so it is observed in nature. And then Z-DNA is a little bit thinner, and it is hypothesized to form at the start of genes as kind of like a locator. And these 3 forms look different, right? Here's A, B, and Z. You can tell when comparing B to A that the A is slightly wider, and when comparing B to Z, Z is slightly thinner. You can see that the room between the grooves is slightly different as well between A, B, and Z. For example, there seems to be slightly more room, especially in the minor groove, between A and B. Here are some additional ways to look at the DNA. This is a space-filling view up top, and you can really kind of tell how A DNA is a bit wider compared to B DNA when we look at it this way. So let's connect our understanding of the A form and B form of DNA to some structure function stuff. So RNA is always in the A form. Why is this? Well, let's recall that RNA ribonucleic acid has an additional hydroxyl group coming off the sugar. That would mean that this hydroxyl group kind of would stick out here in the minor groove. Now, when comparing A DNA to B DNA, you can see how, with the presence of an additional hydroxyl group, you're going to run into some steric hindrance issues with the B form of DNA. So RNA, rather than being in the B form, often is in the A form. What else contributes to nucleic acid structure? The first thing that we can think about is that glycosidic bond, throwback again to carbohydrates. So the glycosidic bond, we're talking about the bond between the anomeric carbon and a non-hydrogen substituent, okay? The glycosidic bond in nucleic acid is in anti. What does that mean? Well, here you see that the syn glycosidic bond kind of lies right on top of that sugar, whereas in the anti, we've rotated that base, so it's kind of pointing the opposite way from the sugar. We always have this anti-orientation to minimize steric hindrance. There's going to be some steric hindrance. If we're not in the anti-conformation. So that contributes to structure in that glycosidic bond is not really going to rotate much, especially in the polymer version. Additionally, folks, one other thing I want us to think about in terms of structure is what's going on with that sugar. We know that the 5-membered ring is not going to be completely planar. It's not really like a boat pose like it is for a 6-membered ring. But for the 5-membered ring, we do have a little bit of puckering where one of those carbons is kind of out of the plane. Think similar to like a half chair conformation or something. The reason for this is because of eclipsing of substituents. If we had this like totally flat 5-membered ring, all the substituents would eclipse one another and there would be steric hindrance. The puckering has a minor contribution to the B, A, and Z conformations. For example, this isn't something for you to memorize, this is just kind of an interesting fact. A DNA has puckering here at the carbon number 3, whereas B DNA has puckering at the carbon number 2. And puckering just between these two different carbons leads to kind of this different shape of the nucleotide that you can see in A DNA versus B DNA. And that different shape at the nucleotide level results in that different structure at the polymer level.